According to the Alzheimer's Association, Alzheimer's is the most common form of dementia. It causes problems with memory, thinking, and behavior. More specifically, Alzheimer's is an autosomal dominant disease that affects brain tissue, creating a negative impact on memory and functionality in otherwise healthy people. This disease impacts approximately 44 million in the world and has two known forms: early onset familial Alzheimer's disease and late onset Alzheimer's disease. Certain mutations in the APP, PSEN1, and PSEN2 genes are known to cause early onset Alzheimer's. These mutations overproduce amyloid beta peptides in the brain and create clumps of amyloid plaques that kill nerve cells in the brain. However, the causes behind late onset Alzheimer's disease are less clear. There is strong evidence to suggest that the E4 allele of the APOE gene increases the risk of this form of Alzheimer's. However, not everyone with that mutation develops late onset Alzheimer's disease. Since 23andMe only tests for late onset Alzheimer's, our SNP chip genetic test will also focus on the late onset Alzheimer's. The 23andMe genetic tests test for variations in the APOE gene. They specifically test for the E4 variant. They do not test for the E2 and E3 variants of the APOE gene. It is established that these variants have no correlation to the risk of developing Alzheimer's. The APOE E4 encodes proteins carrying cholesterol around the brain. The coded proteins are made in brain cells. The APOE E4 allele is associated with an increased buildup of protein clumps, amyloid plaques. These buildups result in the death of nerve cells with transition to the development of Alzheimer's. The 23andMe test has limitations because it does test for variations in all known genes and does not test for the PLD3 and TREM2 genes, which have been shown to increase the risk of Alzheimer's. Our SNP chip will include 3 genetic variations, as there are only 3 variations that have been shown to increase the risk of late onset Alzheimer's. Our test will include the variants on the APOE, PLD3, and TREM2 genes. The variation on the PLD3 gene has been linked to doubling the risk of Alzheimer's disease. In people with Alzheimer's, the expression of PLD3 is significantly lower than normal. PLD3 is expressed in brain cells. PLD3 plays a role in regulating the lysosomal system, myogenesis, late-stage neurogenesis, inhibiting insulin signal transduction, and amyloid precursor protein (APP) processing. If this is expressed in low levels, there can be an increased buildup of protein clumps, amyloid plaques. These buildups result in the death of nerve cells, which transition to the development of Alzheimer's. Our test is better than the test offered by 23andMe because we test for more variations that can increase the risk of developing Alzheimer's disease. We test for the APOE E4 gene, which is the only gene 23andMe tests for, as well as the PLD3 and TREM2 genes. We test for the variants in PLD3 and TREM2 because they have been shown to increase risk of Alzheimer's. In fact, PLD3 doubles the risk of developing the disease. Researching is still ongoing to determine the roles these genes play in Alzheimer's, but research done on patients with the Alzheimer's disease has shown a link between these genes and the disease. Genetic tests for Alzheimer's are not recommended as the result can be hard to interpret. We test for the genes that have correlation to Alzheimer's. This does not guarantee someone will develop Alzheimer's if they carry one of these genes. For example, APOE E4 is carried by approximately 15% of the population, but not everyone will develop Alzheimer's. Despite research that links APOE E4, TREM2, and PLD3 to Alzheimer's, people with these genes will only have a slight increased chance of developing the disease.